Good morning, everyone, and welcome to today's presentation on food and dairy microbiology. I'm excited to have you here as we explore some fascinating topics within this field. Today, we'll be covering a variety of subjects, starting with single-cell oils derived from fungi, then moving on to the value and cultivation of mushrooms, and finally, methods for preserving mushrooms. I'm looking forward to sharing insights and discussing these interesting areas with you. Let's get started. Today, I want to talk about single-cell oil, also known as SCO or microbial oil, which is a fascinating alternative source of lipids. This oil is extracted from various microbial sources like yeast, bacteria, fungi, and algae. These microorganisms are often called oleaginous, meaning they have a remarkable ability to produce and store large amounts of oil inside their cells through a process called fermentation. During fermentation, glucose is converted into ethanol, releasing carbon dioxide and energy, which helps the microorganisms grow and produce lipids. Specifically, fungi are quite special because they can accumulate significant quantities of lipids or oils within their cells, making them excellent candidates for oil production. This single-cell oil production is especially important because it serves as a feedstock for biodiesel, providing a renewable alternative to fossil fuels. Many oleaginous fungi naturally produce polyunsaturated fatty acids, or PUFAs, including omega-3 and omega-6 fatty acids. These PUFAs are crucial for human health, supporting everything from brain function to heart health. Furthermore, because these fatty acids have diverse applications, SCO not only supports renewable energy efforts, but also plays a role in nutrition and various industrial fields. So, understanding and harnessing this microbial oil can really help us move toward more sustainable and health-conscious solutions. Let's take a closer look at the principle behind single-cell oil production from fungi. Essentially, it involves cultivating a special type of fungus known as an oleaginous fungus, which has the remarkable ability to accumulate a large amount of lipids inside its cells, more than 20% of its dry weight. These lipids are stored as triglycerides, or tags, which are rich in polyunsaturated fatty acids. During fermentation, the fungi are grown on a carbon-rich medium, which provides plenty of the nutrients they need to grow. Interestingly, when we limit certain nutrients, particularly nitrogen, it actually triggers the fungi to start storing more lipids. This nutrient limitation is a key point because it pushes the fungi to convert excess carbon into lipids as a storage form. Once these lipids have accumulated inside the fungal cells, the next step is downstream processing. This involves extracting the lipids and purifying them to get a usable product. So, in summary, this process combines specific cultivation conditions with targeted extraction methods, making it a promising approach for producing bio-based oils efficiently. Let's walk through the process involved in developing a microbial product. First, we start with the isolation of the desired microbe, which involves identifying and extracting the specific microorganism that has the potential for our application. Once we have it isolated, the next step is selecting the best strain for production, because not all strains perform equally, and choosing the most efficient one is crucial. After that, we move on to optimizing the growth conditions to ensure maximum yield, things like temperature, pH, and nutrients. Once optimized, we proceed to lab-scale production, where we start producing the microbe in small amounts to test and refine the process. From there, the process scales up to industrial scale, where larger fermentation systems are used. But before going full scale, we often conduct pilot-scale production to troubleshoot and fine-tune the process. After producing the microbe, we move to downstream processing, which includes purification and storage, making sure the product is pure and stable. The next steps are packing the final product for distribution, and then marketing and sales, which are essential to bring the product to the market effectively. This entire process highlights the systematic approach from initial microbe isolation all the way to market, emphasizing the importance of each step in ensuring a successful microbial product. Now, let's talk about how we isolate the fungi that are essential for producing polyunsaturated fatty acids. The fungi we're most interested in include Mortierella alpina, which is known for its high levels of arachidonic acid, or ARA, as well as Schizochytrium species that are rich in DHA. We also look at Mucor species and Cunninghamella species, which are commonly used for producing other poof-rich single-cell oils. These fungi are quite versatile in where they can be found. For example, Mortarella alpina is typically found in soil, making it relatively accessible for cultivation. Schizochytrium species thrive in marine environments, such as coastal waters and estuaries, which makes sense given their marine origin. Meanwhile, Mucor and Cunninghamella species are present in soil, decaying plant material, and even in the air, which means they are widespread and can be isolated from various environments. Understanding where these fungi naturally occur helps us develop effective methods for their isolation and cultivation. This is a crucial step in producing sustainable, high-quality sources of essential fatty acids, connecting directly to our overall goal of developing better alternatives for omega-3 and omega-6 fatty acid production. After we collect the samples, the next step is to culture them on agar media. This allows us to isolate individual colonies, which is crucial for identifying specific strains. Once we've isolated these colonies, we then work on purifying a single, pure culture. This purified culture becomes the basis for our screening process, where we evaluate their potential for lipid content and PUFA production. The ultimate goal here is to select the best fungal strain, one that can produce the desired quantity and quality of PUFAs efficiently. This selection process is key because it ensures we're working with the most promising candidates for large-scale production. By carefully screening and choosing the most efficient strain, we can optimize our production process, making it more effective and sustainable. This step really ties into the overall theme of developing a reliable, high-yield method for PUFA production. This slide doesn't contain enough information. That's why we're moving to the next slide. In the screening process, we use a combination of methods to identify high lipid-producing fungi. First, there's visual screening, which involves staining the cells with Sudan black B. This stain helps us see the accumulated lipids inside the fungal cells clearly. We also observe colony morphology on agar plates, paying attention to characteristics like color, texture, and size, all of which can indicate lipid accumulation. Next, we have lipid content quantification methods. The gravimetric assay is a straightforward approach where we extract lipids from the biomass, evaporate the solvent, and then weigh the remaining lipids to determine the total content. Another method is spectrophotometry, which uses a reagent called sulfophosphovanillin, or SPV. When this reacts with lipids, it produces a colored compound, and measuring the absorbance at a specific wavelength gives us an estimate of the lipid amount. Finally, we use NMR spectroscopy, or nuclear magnetic resonance, to quantify lipids more precisely. This technique measures the area under specific peaks in the spectrum, allowing us to accurately determine lipid content. Bringing it all together, these methods, visual, gravimetric, spectrophotometric, and NMR, complement each other, giving us a comprehensive picture of lipid accumulation. This multi-layered approach is crucial for selecting the best fungal strains for biofuel production, aligning with our overall goal of efficient and sustainable lipid harvesting. Let's start with the fatty acid composition analysis. First, we perform transesterification, which involves converting the extracted lipids into fatty acid methyl esters, or FAMEs. This step is crucial because it makes the fatty acids more volatile and easier to analyze. Next, we use gas chromatography coupled with mass spectrometry, or GCMS. This technique helps us separate and identify each individual fatty acid based on their retention times and mass spectra. It's a powerful method for detailed profiling. Additionally, we utilize high-performance liquid chromatography, or HPLC. This allows us to separate and quantify the fatty acids with high precision, providing detailed information about their concentrations. Now, moving on to the optimization of growth conditions. To maximize fungal growth and intracellular lipid accumulation, we need to carefully optimize several parameters. These include the carbon and nitrogen sources, the CN ratio, pH, temperature, and aeration. Regarding the carbon source, typically, it makes up about 2 to 3% of the medium. Glucose is the most common, but other options like glycerol, xylose, and agricultural wastes such as molasses can also be used effectively. For example, Mordirella alpina prefers about 3% glucose, while species like Schizachytrium can utilize glucose, glycerol, or acetone in a range of 1 to 5%. Similarly, Mucor species and Cunninghamella also thrive on glucose within that range. By optimizing these conditions, especially the carbon source and its concentration, we can significantly enhance lipid production, which is vital for our overall goal of efficient microbial lipid synthesis for various applications. Nitrogen sources are vital nutrients for fungal growth and metabolism. They play a key role because they are essential components of proteins and nucleic acids, which are fundamental for cell growth. When fungi have sufficient nitrogen, it can actually lead to an increase in lipid production, which is often a goal in our processes. For nutrient sources in SCO production, we typically use ammonium salts like ammonium sulfate, nitrates, and organic nitrogen sources such as yeast extract and peptone. These provide the necessary nitrogen to support microbial growth and lipid synthesis. Now, moving on to the CN ratio, which is quite important. A higher carbon to nitrogen ratio, especially when there's excess carbon available, generally promotes lipid accumulation in the fungi. If our goal is to produce lipids, particularly PUFAs, maintaining a CN ratio between about 10, 1, and 40, 1 is ideal. However, it's important to note that nitrogen limitation, meaning less available nitrogen, is usually favorable for lipid buildup. But if there's too much nitrogen, it can actually hinder lipid accumulation and negatively affect overall cell growth. So, balancing this ratio is crucial. Lastly, let's talk about pH. The optimal pH range for these fungi tends to be slightly acidic to neutral. For example, Mordirella alpina prefers around pH 6. Cunninghamella also does well around 6 or slightly higher, around 6 to 7. Schizachytrium and Mucor thrive in a pH range of about 5 to 6. Maintaining the right pH is key because it influences enzyme activity and overall cell health, which ultimately impacts lipid production. So, managing nutrient sources, the CN ratio, and pH are all interconnected strategies to optimize fungal growth and lipid synthesis in our processes. Let's talk about temperature requirements for these microorganisms. The optimal temperature range for many of them is between 20 and 30 degrees Celsius. For example, Mordirella alpina thrives best within 20 to 28 degrees Celsius, so maintaining this range is crucial for its growth. Cunninghamella prefers a slightly broader range, from 20 to 30 degrees Celsius, allowing a bit more flexibility. Schizachytrium, on the other hand, actually prefers warmer conditions, between 28 and 34 degrees Celsius, which can influence how we set up the environment for cultivation. And Mucor, similar to Mordirella alpina, does well within 20 to 30 degrees Celsius. Overall, understanding these temperature preferences helps us optimize growth conditions and maximize yield, which ties back to our overall goal of efficient microbial production.